Hi, my name is Pejman Mohammadi. I'm assistant professor at Scripps Research, computational biology department, and today I'm going to talk about regulatory variation and some of its applications to rare disease diagnostics. So, earlier last year, there was this review of the current state of human genetics research that was published in Nature, which was very nicely showing how over time we started to gain more and more understanding of the genes that cause diseases. So here on the left side, you see from mid-90s, we started to pick up on genes that tend to cause rare diseases. So these are severe diseases that are generally caused by a single mutation that is destroying function of a specific gene. And then as time goes by, we started to pick on thousands of these cases. And with a more delay, we started to pick up on similar signals for common diseases. So this is more subtle. So we're talking about diabetes, heart disease, obesity, normal complex diseases that are not caused by a single devastating mutation, but like many mutations or many variation in many genes or positions in the genome that overall cause to increase propensity to have this disease. And we started picking up on these in the late 2000s, and now we have hundreds of thousands of locations in the genomes that are associated with this kind of health outcomes. Now, this is fantastic. The downside of that is actually in both of these cases, for majority of these findings, for majority of the cases, we're actually kind of lost. And by that, what I mean is that when we look at individuals that we do know they have rare disease, even though we do know all these genes that are associated with them, in majority of those cases, we actually cannot pinpoint in an individual which gene is causing the problem. And I'll talk more about that later in the call. And the same applies to common phenotypes and diseases, and that's that for about 90% of the loci in the genome that we do know they're statistically associated with some sort of health outcome that we're interested in, we actually cannot say why. At the biological level, we don't understand what is causing this statistical signal. And the reason behind that is it's just generally hard to interpret anything that falls outside of the coding genome. So the coding genome is a part of the genome, it's less than 2% of the overall human DNA that corresponds to the parts that produce protein. And for the most part, we have a very good understanding of how that part of the genome works because it's small, it's very well conserved across different animal species, and also it has a specific alphabet. We know each variation corresponds to what kind of change in the protein. We can actually get those mutant proteins and play with them and see how they work. And we know, so we know a lot of things about them. But when we fall outside of that, which is majority of the genome, we're for the most part lost. We don't know what it does. We don't understand the alphabet. We don't even know what that area corresponds to. So for example, if you have some sort of association signal that statistically you found that this part of the genome has something to do with, let's say, diabetes, it's actually not trivial to say this probably has something to do with this gene. In fact, a lot of times it would be some gene that is further away from that is actually somehow associated with the signal. So to solve some of these issues, that is about interpretability of non-coding genome and how variation in the genome corresponds to function, one of the ways that people go about this is to look at intermediate phenotypes. And by that, I mean phenotypes that are not as complex as body mass weight or obesity or heart disease or Alzheimer's and whatnot, but they're much more simple, like expression and activity of a specific gene or the amount of protein that is produced from a given gene. Now, if you could measure this type of intermediate, simple molecular level traits at the mass scale, so for example, measuring all the genes expression in one sample at the same time, And you could do it at some intermediate throughput, like you could do it for 100 people or more. Then you could start to pick up on signals that says, oh, this genetic variant has something to do with expression of that gene. Now, if you also know that this genetic variant happens to also affect the risk for a given disease, then these two together can help you maybe decipher what is going on here at the biological level to basically give you intermediate steps to jump from the genome to more complex phenotypes like diabetes that you're interested in. And so this idea is kind of shown here. For example, let's say if you have let's say this is the statistical significance association from celiac disease, for example, and this is the genome, and maybe this is an area that has something to do with this disease. On its own, it's very cryptic. Maybe this area is somewhere here. You don't know how it works. However, if you happen to have this other plot here that says this area of the genome also is the area that controls the gene expression for this gene, then putting these two facts together, you could come up with a story like, oh, maybe this area in the genome changes the dosage of this gene, and this gene in turn increases the likelihood of getting celiac disease. Now, that's a starting hypothesis, but at least it gives you an actual gene product that you could go there, you could experiment with it, you could increase and decrease the expression, you could do all sorts of things with it. So that's kind of the overall idea. Now, the largest, or one of the largest efforts that has been done to systematically find this kind of signals in the human genome is the GTEx, the Genotype Tissue Expression Consortium that I've been lucky to be part of in the past few years. And so this consortium took about 1,000. They have 1,000 postmortem donors that donated transplant-grade tissues of about 50 tissues. It's not the same across every individual, but about 50 tissues. And what they did was that they measured gene expression for each of these tissues at each of these individuals. And we also did genome sequencing of all these individuals and basically systematically looked for connections between the gene variation in the genome and activity of different genes in different tissues to produce this big resource that can actually tell us how human genome variation works. So, besides this form of common variation and the contribution of gene expression to variation in common phenotypes, we could also use this type of information to actually decipher cases in rare diseases. So I talked about this a little earlier, but rare diseases are generally very severe diseases. That is why they stay rare. But there's so many of them out there, thousands, and combined, they actually affect a lot of people. And the problem with these diseases is that even though they are inherently simple in the sense that they generally are caused with one single flaw in the genome, in majority of the cases, we actually cannot find what is the mutation that is causing it, even though we can easily do whole genome sequencing of the patient and maybe the unaffected parents or siblings. You still cannot pinpoint what is wrong with them because in majority of the cases, you do not have any sort of severe smoking gun mutation in the coding gene. So to address that, we and a bunch of other groups started looking into genes that are somehow dysregulated and say, well, this dysregulation could point us to the genes that are broken. So to do this, we looked at the different ways that you could look at activity of the gene, and that is called allele-specific expression. So to put simply, allele-specific expression is measuring activity of a gene for each of the parents individually. Now, in this case, we don't really care which parent is which parent, but it's just the important part is that we measure it for the two parents independently. And then we use that to compare the activity levels of these two copies. You could kind of think of allele-specific expression as a scale, as a balance scale, it just compares the two things. It doesn't exactly tell you the weight of either of them, but it tells you relatively how it works. And it turns out this is a very robust measure to look at how genes are regulated without getting stuck in all sorts of environmental noise that can clog your analysis. Now, if you do this for all sorts of genes in one person, you're going to look at a plot like this that looks like a cone. Each dot is one specific gene, and on one side you have activity from one copy, and the other side you have that from the other copy. And for the most genes, you're expecting this to fall more or less around the diagonal, corresponding to the case that most genes are active at the same level from the both parental copies. And then if you look at the outside of this cone, you would see some genes that tend to fall outside, corresponding to cases where one of the parent's copies is significantly active at a different level. And so the idea is that some of these things might be those, or one of these things might be the gene that is underlying your problem. So to do this more systematically, we produce this statistical model that basically looks at some sort of reference data like the GTEx data that we talked about to first learn what patterns of variation in a population are normal. And the reason behind it is that our population is not identical. People are different from each other, and that does not necessarily cause disease. So for us to pinpoint specifically the genes that they are way off from the variation that is normal, we need to first learn what is normal. So we produced a model that can learn this statistically and include that with the technical variation to identify if we have an individual that comes in with a disease for each gene, how far from diagonal is too far. Now, when we applied this to a cohort of muscular dystrophy patients, we could see that this method can accurately identify genes that are likely broken in some way in these individuals. So in this case, we have an individual that is already diagnosed, so we use it as a positive control. In green, I'm showing the gene that is actually the culprit gene. And here is our test that identifies this gene, shown as red, with 1 or 2 dozen other genes that seem to be out of control or a little off and dysregulated. Now, these are some 2 other standard tests that can identify genes that generally fall outside of the cone. So what we see here is that these other methods either miss it entirely because they try to be extra conservative, or find it, but they find hundreds of other genes with it, which really decrease the value of it because you can't just follow up 400 other genes. So that was great, and we could use that to identify some of the patients that we had previously not diagnosed. So one thing that we thought was that, okay, now let's just apply this to general population, because general population people also have a lot of rare variants. Many of them are functional. They do not cause rare diseases. That's why we call them general population. But they still can have a variant that makes them significantly taller than what they're otherwise you would expect them to be, or significantly more obese or more lean or stuff like that. And generally identifying these sort of functional rare variants is a hard task because by definition they're rare and you don't have that much statistical power to get an understanding of what these things do. So what we did is that we banded over a couple other groups and we looked at all sorts of phenotype signals that we could get out of standard RNA-seq data and see how can we use outliers in these signals to identify variants that are likely doing something. So what we found is that all these signals do indeed help to find rare variants, although each of them point us to a different class of rare variants. And if you actually include these things in a probabilistic model, you could try to pinpoint what are the likely variants that are underlying these. And actually, if you look at other data sets like in UK Biobank, we could show that these rare variants that we think actually they're functional, they tend to have a larger effect on common phenotypes than an average variant interest. But another thing that we were interested in when we were looking at this data was how good do these outlier signals replicate across different tissues? And this is an extremely important practical question because in practice, when you're looking, let's say, at the patient in a clinic, or when you're just trying to put together a consortium to look at, let's say, some sort of signal across many samples, you likely are going to only access things like blood. Now, if you're lucky, maybe you have a skin biopsy or muscle biopsy or something like that. But generally, you don't go to people and cut out random tissues out of them. So what is very important, especially in the clinic, is that you would be able to identify genetic variants that are of interest from tissues that are accessible to you. And so what we did here is that we looked systematically in GTEx data, how good can we replicate the stuff that we find in other tissues from other tissues. So in these plots here, you see the replication between all possible pairs of tissues in the GTEx, and the more red they are, the more replicability we have. So a couple of things that you could see in here is that first, all of them are to some extent red, so there's some level of replication going on. There is gene expression is more pale than the others. ASC is the most red one, so that is generally consistent with our understanding of the noise in gene expression and robustness of ASC data. So that's good, and we were happy because we were the ASC people. But the thing that is still a problem is that even for ASC, where we have the strongest replication signal, still these numbers hover around 20 to 30%, meaning that when we look at one specific tissue, we only find we basically still miss 70–80% of the hits that we would like to see if we were looking at some other tissues. So with that, and that remains a big limitation. So with that, I'm going to stop my talk and tell you briefly about what are the things that we're doing to address some of these questions. So now we're looking to apply some of these methods for improving the yield of rare disease diagnostics to a range of different cohorts, including neonatal cases of rare diseases, neuromuscular disorders, pediatric cases of heart disease, and also a general cohort of hard to diagnose cases that is put together in the Undiagnosed Disease Network by the NIH Common Fund. But besides that, we're also launching a lot of efforts to get around this problem of identifying variants that are of interest from tissues that are accessible. So that involves improving things statistically by improving statistical power through a range of different methods that can identify dysregulated genes more accurately from the data that we have available to us, but also looking into methods that can help us identify which tissues are more helpful or what kind of genes are likely causing our disease, and also looking at sort of biological and pharmacological interventions that can help us to access genes that are otherwise not accessible from tissues that we have at hand, like in blood, and see, is there a way we could modify these cells somehow to reach genes that we would not otherwise have it normally. So with that, I would like to thank everyone who has been contributing to some of these works that I've been presenting. So my lab at Scripps, but also our collaborators in New York, specifically Julie Lapalainen's lab in New York Genome Center, which was a postdoc advisor, and some of these work started and matured there. But also our collaborators at the NIH and the Radies Hospital that are helping us with the clinical work, and at the Broad Institute, we work together on the rare disease of the muscular rare diseases, and Stephen Montgomery and Alexis Ballard's lab at Stanford and Johns Hopkins that we work together on the rare variants in the GTEx Consortium. And with that, I would like to thank everyone for joining this virtual talk.